Thanks everyone for being here, either in person or on Zoom. So it's truly a pleasure today to welcome Dr. Robyn Tanguay. Dr. Tanguay is a university distinguished professor in the Department of Environmental Molecular Toxicology at Oregon State. Also serves as director of both the Superfund Research Center and the Hoover Aquatic Research Laboratory, one of the world's leading facilities for high-throughput blood toxicology research. She earned her BA in biology from Cal State University, her PhD in biochemistry from UC Riverside, and then did a short postdoc at UC Riverside, and then the majority of her postdoc at University of Wisconsin-Madison. Over the course of her career, she's become internationally recognized for pioneering the development of high-throughput zebrafish screening platforms to evaluate and predict the toxicity of chemicals in complex mixtures. Her work has fundamentally advanced how we assess chemical hazards, bringing greater speed, biological relevance, and predictive power to environmental health sciences. Beyond her scientific innovation, she's deeply committed to training and mentoring the next generation of scientists. And her leadership will certainly help shape the broader field of environmental toxicology. I could go on and on. Presidential chain of SOT, more than 320 papers, over a dozen book chapters, but we'd probably be here all day, so I will leave it at that and say we're very fortunate to have you here today. Please join me in welcome. Thank you, Chris.
Good afternoon. Really appreciate the invitation. I know you guys are all busy. I'm busy as well, but what I like most about this job is actually getting to meet people in their natural habitat. One thing I want to say for like the trainees who are here, and Melissa and I had a talk this morning, and I think it's a tough time to be a scientist in America. Right? It's tough to be an American in America right now. But I can't emphasize how optimistic I am about where science is. Because if you think about, if your goal is just to get a job, you can do anything, right? But if you get into this business because you're trying to solve big problems and advance the world in some way, we are so well positioned to do that now. And it's unfortunate that there's things getting in the way, being able to implement all of our capabilities to solve it. But I think we got to keep at it. And I think it's a really exciting time. And I'll talk a little bit about the way we're trying to contribute to this. I want to start with acknowledgement. So I have a huge group and lots of different structures. So we have Lisa Trong is my deputy director. She's a former student and now she's a research track faculty. We have an engineering team that we keep retooling. So some of the instruments I'm going to show you, we invented, designed, and implemented. Really good funding from NIEHS and other sources. We have collaborations at NC State, University of Washington, and OSU is part of our Superfund. I collaborate with everyone there. And then really importantly, our Superfund is a collaboration between Oregon State University and the Pacific Northwest National Lab. So Katrina Waters, my deputy director, and then Ryan and others. And we work with a lot of companies, and I'm going to talk a little bit about two of them today, Cedar Sciences and Orgona Optics. So when I think about, when I put these talks together, someone asked them, one of the students asked me this morning, like big picture thinking versus little. So I think I'm always a big picture thinker. And so I try to think, what are the core challenges in environmental health sciences? And then some of the rationales and why would I choose to use this particular model to try to solve some of those challenges? And you do have to make a justification for that. We all do. And then I'll give a couple examples. So it's going to be observation. So when you sit in an airplane, you just stare out the window. So the challenge is this is what toxicology is. This is what drug discovery is. This is what material science is. We do not fully understand how chemicals and their mixtures interact with biology. And if anyone says we do, they're overstating their knowledge. And really the basis of that is really our insufficient understanding of biology. So I go to a lot of NAM conferences around the world, and with all the confidence in an organ and a chip and organoids, and we got this solution. And some of those advances are remarkable. I don't dismiss it, but to think that those replace the integration of complex biology and we can predict the adverse outcome of a chemical in those systems, we just can't. And in fact, why do we do animal testing? literally because we don't know what's going to happen if we do these exposures. And then we were challenged to look to see if we could see what happened. And certainly there's many animal systems can be replaced for some questions. There's no doubt about it. But if you ultimately want to know whether a chemical is going to adversely affect the whole organism, I am confident we will be able to do that someday. I don't think we're there yet. And so what are we good at? We're exceptionally good as human beings at making chemicals do what we want them to do, right? If you're trying to make a drug, rational drug design, find a target, find a chemical that binds tightly and with kinetics that inhibit or activate the pathway. We're really good at that. Material science world, you want a chemical to block UV rays, we can do that really well because we have the selective pressure, the function drives the selection of that chemistry in those materials in those examples. And that's a good thing. So we're really good at efficiently getting to a point. Honestly, in very few circumstances do you actually put the potential unintended consequences in the same concern, right? It's an afterthought, right? So when you think about developing a drug, oftentimes, I mean, it is improving. The NAMs in drug discovery are really impacting the pipeline right now. But you'll do this really, you get it to this stage of development, the next stage, and looking good, okay, now get it to the safety people, right? And we're losing so much time and money because we're not putting safety at the beginning. And a lot of that is because of the cost. It's the cost and the speed of our traditional approaches that are limiting that innovation that's on the front end of that discovery. So if you could start driving those costs down, you could put safety side by side with product development. So I think that's what can happen. So ultimately what we want to do, can we predict a biological activity solely based on the structure of a molecule? We can't yet. QSAR type approaches kind of gets there a little bit, but only that really, when you think of a QSAR, you have a specific activity and you can predict the chemical effect on that activity. That's pretty good. But when you have these new molecules, they might actually start hitting other targets that you don't see in your systems. So I do think NAMs, new approach methods, not non-animal model, new approach methods will, I think, provide that earlier selective pressure for safety that'll advance. So when we work with companies, that's kind of the message. It's not like I'm a toxicologist, I'm here to shut you down, because that's what they think all the time. You meet a toxicologist. Instead, it's like I'm a toxicologist. I'm literally here to help. And that's kind of, and that goes better, as you might imagine. So understanding chemical bioactivity. So everyone has a different context where they work. But when would it be important to know the activity of them or being able to predict the activity of chemical? Drug discovery, obvious. Legacy chemicals. Still, there's a need with limited resources, say cleaning up sites, reducing human and environmental exposures, identifying which chemicals we should prioritize our efforts. We need to do that better. We cannot get to zero level exposure to all our toxicants. And public may want that. That's not going to happen. So how do we actually make better decisions with the limited resources? Chemical mixtures. Everyone says, oh, we have a mixtures problem. We do have a mixtures problem. Some of these approaches could help deconvolve some mixtures problem. We work a lot with exposure reduction technologies, the Superfund and elsewise. Can we use these tools as guidance for whether or not these technologies are actually making the situation better or worse? And of course, providing information to green chemistry. So there's lots of times where a decision maker needs to understand chemical activity, and it's not just in any one of these boxes. So with all the excitement about new approach methods, so I've been on a number of these National Academy reports, and so some of the recommendation of the last two is, and everyone's just buying this. I'm not. That we need to go into these simpler systems. I understand the motivation, cost, speed, etc., and to use human biology. Right, that's where they lose me. Right, there's an assumption that when you take this simpler system, no longer in a mouse, it's no longer a rat, it's not a fish, and you use human genetics driving this simple system, that's going to be more predictive of a whole animal outcome than if you would do a whole animal outcome that has genetics based on, say, mice. And I don't think there's any evidence to indicate that's a true statement. Right? So I say if you're developing a drug to hit a human condition, using the active site for a human gene makes a heck of a lot of sense. That's not the same thing as doing safety assessment for a chemical in a whole organism. And that's the divide. So when I go to these talks, it's like I make them think about where you would convince me. Say you took all this human NAM development and then do the exact parallel development of mouse NAMs. Mouse on a chip, mouse organ on a chip, mouse organoids, and then see which ones. My guess is they're going to agree a lot more similarly than the disagreement that we see from using a mouse in say human experience. So, and the reason is because you're balancing the importance of genetics, which there are certainly times where the genetics really dominates, but the biological complexity when you have a chemical exposure to a whole organism. And I would argue the cell-to-cell, organ-to-organ communication, chemical transformation, all of that is vastly more important than the origin of the genes that are giving rise to that organization. And that argument is being lost in this discussion of NAMs. So we proposed for a number of years that the zebrafish embryonic development in general is really a good opportunity to identify hazardous chemicals and their mechanisms. So this idea that I think the biological complexity, at least our state of knowledge, is more important than using the genome of a human to make these decisions. So just like 3 weeks ago, the EPA announced again the 2035 deadline, but this time they were very careful with their language to reduce the use of mammals in chemical testing. So our system is actually could be useful still, which is good for me. So why embryonic development? So again, I had a choice. I'm not a fish person. I wasn't trained. The question this morning, I was not trained by a fish lab. I had to make a decision as a postdoc where I wanted to tackle big problems and how I wanted to do it. So I decided to develop the zebrafish model. Because when you think about a chemical cannot cause adverse effects unless it modulates an activity of something important. That's toxicology, right? And that's going to be route of exposure dependent, life stage, organism. But we know that during embryonic development, that's where we're most susceptible to chemical insult. That is widely recognized. And it's because of the fundamentals of developmental biology. That's when the most dynamic, the programs are being acted upon to make cell types and organ types and cell-cell communication and all those wonderful mechanisms that developmental biologists have worked on for a number of years. And then it's also when we're most conserved, when mice are most similar to human and most similar to fish, because those processes in development are so well conserved. And more operationally, most of the genome in an organism is called upon to pull some activity off during development. Sometimes it's a very short period of time, right? So it's like when I talk to my cancer biologist friends, there is no such thing as a cancer gene. Can't say that in an oncology department, but in fact, these are developmental genes that are perturbed that cause cancer, right? So that's an operational way to saying, well, if a chemical, an unknown chemical, a new mixture is going to cause an adverse effect, it must modulate something, and that something will be expressed with high probability during embryonic development. So this then becomes a biosensor for all chemicals. This is kind of how we operationalize. So that's the concept. So we wanted to say, what would we look for? So we want to be able to tell a normal animal for an animal that's been perturbed in some way by your exposure condition. I'll show you how we did that. So what we've done over the years with a lot of deliberate thought. So first we wanted to develop had the conversation about having a genetically diverse background. So a lot of the zebrafish labs and mouse labs and Drosophila labs, they get almost monogenic, very, very narrow genetics colonies or groups of animals. And I wanted to have a genetically diverse strain. So we went to a place in Florida about almost 20 years ago now, where they raise these fish by the billions of fish. So lots of genetic diversity. And then I created that and made a pathogen-free colony out of it. So we sequenced this genome hundreds and hundreds of times. So it's like 4 to 5 times more diverse than any of the zebrafish wild type whites. That's a pain in the rear end for some molecular work that we do, but it's great when you're trying to get a diverse response to chemical exposures. And I'll show you, we fully automated all of this to improve the rigor at which we can do it and the reproducibility while also lowering costs. These models will only be used if the cost drops. So dropping labor costs and increasing reproducibility and et cetera. So what we are able to do for every chemical a new chemical, we have different stages of testing, but ultimately the definitive testing, we do 32 replicate animals at every concentration and 11 concentration. These are in vivo studies. So this is something that you can't even come close to approach in any other in vivo context. And then allows you to get these curves that the EPA loves, benchmark those curves, et cetera. And what we've done many, many years ago is we've identified a number of endpoints that you can measure reproducibly, cost effectively, that really captures broad bioactivity. So that means our screens are pretty agnostic to the chemical, which is important. Because again, we're probing biology with chemicals, so it should be agnostic to the chemical. And we'll see these endpoints frequently from chemicals that sometimes we know the mode of action. We know the molecular initiating event, we know the entire cascade. We've worked some of those out ourselves. And they produce the same phenotypes from another one that enters biology a different way. And it's because this complexity of development is so interdependent on so many networks that development kind of collapses in a common way. And then the embryo just reveals that an adverse effect happened. So then when we started doing this and finding statistical association between all the things we're measuring, we can strip back what we have to measure to accurately identify bioactivity. And then we also have behavioral assays that we kept at the same time. So our timeline for our normal high throughput screening is from, we go from the 10 minutes after fertilization. So at 6 hours post-fertilization is when we initiate the exposure. And typically, not always, we typically do continuous exposure. And we're just challenging the animal to develop normally in the presence of graded concentrations of this chemical. And then we have measures that we do at 24 hours and we do nothing typically between 24 and 120 hours. And we measure a number of these phenotypes. We have an early embryonic photomotor response assay, and we do mortality detection at 24 hours. And then we have the 2 behavioral assays and then morphology screens. I'll show you how we do this. So we just finished this wave of renovation. This is done now. So this is a fully automated. I'll walk you through it real quick. This room is like 28 degrees. Our equipment does not like to stay hot, but the embryos have it. So everything from the beginning to the end, we had to reimagine how you do this in an aquatic situation to get the throughput. The way we raise the fish, the way we collect the embryos, for example. The first thing you did, we have these, we have like 30 of these tanks where we have hundreds of fish in a tank, and we could put a collection device. So when the lights come on, they spawn over this basket. And thousands of embryos come in, and we can just collect them using a little embryos on tap, basically. So one of those tanks, we get 8 to 20,000 embryos in less than a minute. That would take hours to do in the past. Again, it's not just time. It's just when you're able to do these studies at scale, you absolutely do different studies. Your imagination is kind of great. Then we created this instrument to remove the eggshell, the chorion. That's just kind of what it looks like. And so after 14 minutes, the eggshells are completely removed. And then now the embryos are really fragile. In this station, so this is, we have station automatically barcodes these plates and shoves it back into the stack. And then when we're ready to go, we have the robot pulls them out. We fill our plates with embryo media. And then when they're full of embryo media, then we have to use the robots that then can individually pick up these embryos and put them into a multi-well plate. And this is done in about 6 minutes per plate. And then after we fill the plates, it goes to the digital dispensing. It's a Hewlett Packard, so it can dispense like picoliter volume, so we can hit really, really, really precise concentrations of chemicals, like 0.05 micromolar. Sometimes we need curves that steep. And then after that, then the plate have to get sealed. We seal all of our plates. We don't get evaporation as that goes into there. And then after the plate is sealed, it goes into a stack. And then it sits there for 24 hours and then it goes into our first behavioral unit. And basically what we're capturing this spontaneous motion that fish do starting at about 18 hours. We know the whole circuitry that controls that. And then we blast it with bright light. So we capture most of the activity in infrared light. And then you get this rapid tail twitching. And then we do it again and we typically don't get a response. That's a really, really earliest indication that some aspect of development has been perturbed, just capturing that behavior. Then it goes back to the stack. That 24 hours comes out of the stack. Another robot picks it up. The barcode scans it. And now it has to decide which one of the 9 behavioral units that is going to receive it. First, you have to take the seal off, which is actually the hardest part. So we deseal the plates and then it spins around, throws it into the zebra boxes. And then we capture the larval photomotor response and the startle response data from every single animal in every well. And then after that's done, it puts back in the stack and then it picks it up and puts it into our imager. So we work with Ramona optics. And so normally when you're working with the microscope and these embryos, you put on like a 2x objective, you can see the entire embryo in one field of view, but at really low resolution. So if you wanted to capture an image at higher resolution, you may have to take 20 images of every well and stitch them together. If you do 50 to 100 plates a day, you literally can't do it. It takes forever. So with Ramona, they have 54 cameras and 54 lenses simultaneously looking at your object. So you can image an entire plate in like a half a second at very high resolution. So that just revolutionized how fast we can image it.
So can I,
So we can, yep.
one endpoint per well?
No, all of these endpoints are captured in the time course of the time of development. So that means every well is you measure 2 behavioral assays, 3 behavioral assays, and then our morphology and malformations are all done in every well, which is in our LIMS system. We know exactly what chemicals in that well, what concentration in that well, and all that data is automatically extracted using machine learning. And then all that data is analyzed like within 10 minutes after that last assessment, all the data is done.
And is there a failure rate for getting development of the embryos?
Yeah, there is. So we have a QA criteria, like 20%. If our controls are off by 20%, the entire experiment has to be replaced. And so our noise down to 20% is where we put it. A lot of that is, some of it is a background from the decoordination. So if we didn't decoordinate, we could probably get it to 5%. But it's definitely worth it for the measures to get that coordinate out of the way.
Thank you.
But we've used this for tens of thousands of chemicals and mixtures. Examples are listed here. So for the Superfund, I thought I'd just give this example. So one thing that if you pay attention to this NAM space and the EPA and regulatory agencies, they do a lot of chemical read across, right? So they know something about this chemical and they find chemicals that are similar to each other. And let's just assume they all behave the same way. And then you could sigma your concentrations and predict an outcome. I think that's a really dangerous thing to do. And I'll show these examples will show that. So we know certainly the pHs, we know that they have at least 2 aromatic rings fused. They get substituted as well. If you go in the environment in our Superfund, Kim Anderson's group and Stacy Simonich prior to a few years ago, you find hundreds and hundreds of different pHs, whether they're hydroxylated, nitrated, et cetera. Very little data on these. So most of the priority pHs are focused on cancer as an endpoint, which is probably appropriate at that time. But we're not even measuring the pHs that are at the highest concentrations in the environment that humans are exposed to. So that's a problem. That's what our center has made our regional area EPA office very uncomfortable, because we're publishing that, if you go to Portland Harbor, some of these hydroxylated pHs are 1,000 times higher than the priority pHs. No tox data, right? So that got people upset. So we know that some pHs engage with the H receptor to either induce metabolism or toxicity itself. But we right now could not look at a pH and predict with any certainty the toxicity. we might be able to predict some of the metabolism and the likelihood of being a mutant or mutagen. So that's what we're trying to fill that gap. So Kim, this is a couple of years ago now, our first library had like 126 pHs. And everything we do in the lab, everything we're blinded to. We have no idea what the chemical is until after those 5 days when we revealed the key. So she gave us pHs anywhere from 2 to 7 rings, and they could be the parent pHs, these are the more common ones that the EPA measures, or these various modified ones, heterocyclic and methylated, et cetera. And then we just said, let's run them in a single system. So the only thing that varies is the chemical structure, right? Of course, absorption and that's all in play, but one of the biggest challenges that say EPA or National Toxicology Program, et cetera, are trying to, when you try to take data that's been collected in different systems and then make a decision, because you don't know enough about any system and they're all different and the experimental conditions could impact the results. But when you actually have a single system, everything is controlled. You can say this chemical behaved differently than this chemical with high confidence. That's what we try to do. So again, for this one, at this time, we were doing 22 morphological endpoints. These are examples that are illustrated here. This is that early embryonic photomotor response assay I told you about. You get the spontaneous motion, and then you hit it with bright light that they actually can, or they have photoreceptors in their hindbrain. Their eyes aren't functional yet. And then it gets lots of activity. And then as you increase the concentration in this example, the activity goes down. And then this is a larval photomotor response assay. We just turn the lights on and off and measure their activity. And then since we are interested in the potential role of the H receptor, we implemented two different ways of measuring activation of AHR. We've done high throughput immunohistochemistry. And then also we have a reporter line that has GFP driven by the CYP1A, the Zebrafish CYP1A promoter. So you can visualize it this way. These are not exactly recapitulating each expression, but it's pretty similar. And even when we did this, we would see different patterns that we've never seen before. We saw examples where pHs that were toxic, there's no activation of CYP. That now, some where only the liver is induced. We have some where the skin and neuromass are expressed, some just the skin. And then this is more typical, like if I expose these fish to dioxin, this is what you'd see. You get this vascular expression of HR activation and CYP expression. So this just, all of this information, we want to just track it. Is there a relationship between the structure and this collection of activity in the single system? And this is kind of what you get. You can start breaking them down based on, these are the morphology endpoints. So anytime you see a blue box, that means we saw this activity at a really low nominal water concentration. And where we don't see anything, that means from low to high concentration, this chemical did not affect that response. And then we have two columns for behavior. We got the embryonic photomotor response assay, and then the LPR is here. And then we also track, is cytochrome P450 induced, and where is it induced? So how can we bring all this information together? So you start clustering it based on the phenotypes, and you see this big cluster up here. These are all really toxic at low concentrations, suggesting that perhaps these may behave through a similar mechanism. And you go on the other extreme down here, these pHs from low to high concentration, absolutely nothing. And then as you go up the chain, we have some where it only affect one part of a behavioral response, and some both behavioral responses. So there's a whole mix of activities that we see when we control the situation. So we do this. So we've done 386 individual pHs now. So I don't have a heat map big enough to show you that right now. But it's still not enough. I talked about all these endpoints that are with these common endpoints from different mechanisms of insult. And that's not very gratifying, particularly if you're used to, like, I want to screen for, say, endocrine disruption. I want to know whether the estrogen receptor is activated. If you activate the estrogen receptor, you get some of these endpoints. But these endpoints don't tell you you've activated the estrogen receptor, right? So we use transcriptomics a lot. So we use phenotypically anchored transcriptomics. And the reason is because these common endpoints and again, different key events can produce the same effect. So the EPA is going hard on using points of departure of transcriptomics to make chemical decisions, or using patterns of transcription responses to do chemical recross. So we're trying to contribute to that, but doing it in a whole animal system. So we do it a little differently than some folks. So we want to measure the transcriptomic responses that might be causally related to the endpoints we measure. And most of the endpoints are done at 120 hours. So what we do our concentration response curves, measure all the endpoints that I talked about, find the concentration response curve, and then we want to back up, repeat the experiment, back up and do that exact experiment and collect RNA when the animals are normal looking. Right, so we think like kind of you open up the hood, well, it's starting to get messed up inside, but the animal hasn't revealed it yet because development hasn't progressed. So then we wanted to use this as a kind of a single slice for biologically active molecules to measure what's happening transcriptionally. And so we're doing an individual chemical, that's fine. You can do this across development. But when you start taking thousands of chemicals, you start asking the question, are there conversion points in development that might explain why these endpoints end up being very similar? So that's what we're trying to do in process. So we pick the time point and we pick 48 hours because it's where most of organogenesis is done, right? Most of the chemicals, when we do windows of susceptibility, it's usually between 0 and 48 hours. It seems like not all, we got burned a few times. And so 24 hours, it's just too dynamic. So the data was a little bit less clean. 48 hours is kind of a little bit before the liver is present. So that's where we get into trouble, where some chemicals that are metabolized, like here, we have signatures that are missing. So then the question is what concentration, right? You know, it's getting cheaper now. We can do it across concentration, but if we had to pick a single concentration, we pick the EC80. That means that 48-hour time point, if you sampled it, 80% of those animals will be affected at 120 hours. They're all normal now, so you can't identify them. So we want to pick a pool response will have a very high signal to noise in that pool. So that's why we picked that concentration. So we picked 16 pHs from that list and then ran this analysis. What we found is this one group with cluster B, they shared a lot of transcriptional signatures and pathways, suggesting that they may share maybe an initiating event or some mechanism. But even with that, some of these had unique transcription problems. This is at 48 hours. Remember, all of these animals look completely normal, but they're being modulated in a huge way. So we have other data. So we can explain these. We can do HR knockouts, and we know that the toxicity, each one of these are completely HR dependent. Some of these unique transcriptional profiles we think is due to metabolism of that compound and modulating other targets. So we've used this in PFAS and other molecules as well. So diving a little bit closer, so routine. So we worked with Kim Anderson. So this particular pH that you see here, not one of the priority pHs that the EPA is monitoring. They don't do any alkylated pHs. And when we measure this in the environment, it is the highest abundant pH for human exposure and in aqueous environments. So we get it, they're certainly from industrial processes, but from like forest fires, like this one that hit up in Oregon, Washington, concentration just incredibly high. And so our center wanted to contribute to what do we know about this compound. And we learned a lot from chasing this guy down in the center. Since it's not a measure, now Kim has done passive sampling. I'm sure you guys know about the passive sampling devices that Kim Anderson deploys. So if we went to the Houston area where there's a lot of this going on in Houston, if you've been there, you can smell it. Lots of chemical and petrochemical companies. So everybody's exposed to routine. And even in New York, 54% of pregnant women in this cohort that they're studying had high routine concentrations. So humans are exposed. When we run routine in our screen, like I described, we get this in the wild type animals, you get this, concentration response curve. And if you do this in knockout, it's largely dead in the water. So literally, this is a routine exposed. This is an HR2, one of the HR2 lines that we made. And these animals are completely resistant to the same concentration. So this toxicity is 100% HR2 dependent, and these are the endpoints that we track. So clear, it's an HR2 ligand, just like TCD and other pHs that we studied before. So we thought. when we actually do a concentration response, DEGs, what we wanted to determine is trying to identify which increase in gene expression is causally related to this in vivo response. And when we do that, we did transcriptomic for a number of concentrations, very low concentration to high concentration. Certainly, the higher their concentration, more DEG starting occurring, but there's a whole lot of adaptive probably responses are happening when there isn't a phenotype. But interestingly, some of the same genes like CYP1A, their magnitude goes up and more transcriptional changes. So this doesn't resolve it completely, but certainly when you hit these animals with a strong HR ligand, you get an adverse outcome. But if you did it at a lower concentration, the animals could recover from it. Bottom line is. So when you get these DGs, this is what my river grant is really focused on: screening at least 10,000 compounds across concentration, do transcriptomics on the hits, and then try to modulate the targets that are identified from those studies. But it's easier said than done, right? At scale. So when you get a differential expressed gene, if you're studying a simple system, if you study one thing, it seems simple because you're blind to everything that's happening. But if you start doing everything in a systems approach, it's like, it drives you crazy. But that's the only way we're going to solve this complex biology, is by caring about their biology. So if you get a gene expression change that seems causally or temporally and spatially linked, it may be necessary for toxicity. It may be an adaptive response, or it may just happen. Right? Sometimes it's connected to another gene and it doesn't matter. That's not very useful biomarker. So when we did CYP, so when I was a postdoc, I wanted to do this and we just didn't have the tools at the time. So, so when I was trained, CYP1A induction is the cause of dioxin toxicity, right? Really? So I kind of believed that for a bit. Until I started doing more stuff, like, no, it's not. I mean, it's a really good biomarker for activation or exposure, biomarker for exposure. So, and it's been demonstrated now in mice. So what we did is we created 2 different knockout lines where we knocked out CYP and wanted to know how important is CYP for the response to some of these new pHs that we've identified as HR dependent. So, and interestingly, we had one that created a stop code and one that put a 5 amino acid insertion. That was interesting to chase that one out too. So first we did dioxin, had to do that experiment. So essentially when you knock out CYP1A, you still get all the toxicity that you see. So it's not the induction of CYP that's causing toxicity. It's all those other genes that are being modulated by TCD that's causing toxicity, which is what we've been showing. So that made it definitive. Interestingly, routine is different. So this curve is the wild type. And when you knock out CYP1A in either one of these knockout lines, the animals become way more sensitive, right? Suggesting that CYP1A is responsible for detoxification of routine. So that seemed like a nice clean system. And we went a little deeper. And then we said, why hasn't anybody studied this in all these, like ToxCast hasn't touched routine. They didn't, no one's studying routine. So we worked with Indigo Biosciences, Jack Vandelhaven's company. And so we grabbed a bunch of pHs that we've studied, and we have the knowledge of whether it's HR dependent, et cetera, and ran it in their system. And it's a reporter gene system where the HREs drive in luciferase, really sensitive system, really great platform. And when we throw routine in here, it's dead in the water with zebrafish cells. It's also dead in the water with human cells. Even though everything indicates that routine is an HR2 ligand, it does not activate these heterologous systems. Whereas some of these other pHs we identified certainly do. These are all HR dependent, the other ones, and they're all active. And the correlation that we see with known human HR ligands and zebrafish HR2 ligands, 1-to-1. We do not see any differences. So these receptor, the binding sites are really well conserved. So something funny is going on here with routine. So our hypothesis is we don't think this is the primary target. We think it's metabolized to something, and then it becomes a target, and then it's detoxified by CYP induction. So this is not easy to do in vivo, but really small in vivo. So what we found is first by just measuring the body burden. So just measuring routine. This is Kim, our chemical analysis core helped us with this. We see that we get accumulation of the parent, and then it starts to drop. And then when we start looking for the appearance of various metabolites, then we see they start appearing at the same time. So it is being metabolized. If we use this reporter line, and if we look at PKF and induction, So early on in 24 hours, you see the GFP signal come way early and really, really bright. And routine, nothing, you just start seeing it pick up here and here. So this also corresponds that we're creating a metabolite from some probably early hepatic metabolism that creates this HR ligand. So what we really think that the HR2 dependent toxicity begins at 36 hours, and we think it's through some metabolism. We have not identified the actual metabolite yet. We can dock these things, but honestly, routine docks pretty well too. So it's not easy to explain, but the bottom line is when I talk to folks at the talk guest, most of the systems, almost all, not all, but almost all of these simpler systems lack metabolism. Now imagine you're developing a product and you have a panel of NAMs that you can select. Human genetics, great, right? It's better. And then you get the go ahead because you have no secret, but you don't have metabolism. Some of it, predicting metabolism is getting better. That's not perfect either. So there's still a role for a whole organism, I think. So now we have folks looking into this system and some human systems, more intact human systems. But then, I said that our assays are agnostic. So the biology is agnostic, but that doesn't mean the exposures are not problematic. So these small molecular weight molecules, volatile and semi-volatile compounds, testing them in any system is really challenging. Particularly NAMs in a dish, for example, they all volatilize. So we studied a bunch of naphthalines in that initial dump of chemicals that Kim gave us. We know that alkylated pHs are a lot more dominant in the environment, including alkylated naphthalines, and nobody is measuring alkylated naphthalines in the environment. There's tons of it. Again, EPAs, they categorize it, they have 2 rings, put it in this bin. So I think our data is showing it. So when we did this originally, most of these alkylated naphthalines in our screen, after we revealed the key, were inactive, right? So again, we didn't know what they were. We didn't know they were volatile. So we just, so now when we get a client, we have to handle this differently. We get a client, they know something about their molecule. They can't tell the screen team what the molecule is. They tell somebody else. But when we understand the phys-chem property, then they tell the screen team, well, you may want to use this different approach for this chemical. This is a smarter way to go. When we did the ToxCast screening 15 years ago, we couldn't do that. We just had an ID. Anyhow, so we ran this and very little activity. So these are all the alkylated naphthalines, and only 2 of them created an effect at a pretty high concentration. These are the EC effective concentration that cause a significant effect, and just a behavior loss. They're mostly dead. We know this is due to volatilization. We developed a dosimetry model recently that proves that. But I think these molecules need to be evaluated. There are tons of human exposure. How can we test in the system? So we did. I had Mackenzie. We put 8 embryos into a glass vial. That doesn't solve it. If you have any headspace at all, it's still going to go to the headspace. If you have a membrane above the headspace, it'll stick to the membrane. Right? So how do we keep the chemical available for the molecule? So we put these 8 of them into a vial, and then we 3D printed this little wheel that put into the old hive ovens that some of us use here. And then so the guys are tumbling, and the embryo is put in this direction. So as this thing rotates, the embryos are constantly, so the headspace can't be established. So the embryo keeps bumping into the molecule. We said, will that work? So we ran that. And then the way we did it, you can't tumble a developing organism without for 48 hours. They don't like it. But they can handle 24 hours, no problem. So after 24 hours of exposure, we then pick them up, robots put them into the plates, and then we assess them individually, just like I described. And what we found is suddenly these molecules that were completely inactive, some are very, very active, and some have very low concentrations. And interestingly, the parent compound, naphthalene, inactive, and even some of the compounds that are measured in urine, for example, are inactive. But some of these down here are really active at really low nominal water concentrations. So we for the last year and a half or so, we've really been trying to leverage, see whether or not we can use some of these new tools of AlphaFold predictions of structures to add another dimension of data to our in vivo and structural information. And so basically in this system, and this is just an early example, we take the AlphaFold prediction of 16,000 human proteins and 38,000 binding pockets. So some proteins have more than one. And so you get those coordinates, you get this massive database, and then you can take every molecule that we have that we already have tox data on, differential tox data, and now we can get this predictive interaction score. Most of it, totally agree, is a bunch of baloney, and it's not going to be biologically relevant. But when the baloney starts to align to the data we have, there may be a signature there to chase down. So that's what we're trying to do at scale. This is not fast to do, to take, but we have really fast computers now, so we can even pushing thousands and thousands of chemicals through this approach. It's been really fun. So this is an example. So we take these alkylate naphthalene, so the structure, the names are down here. And what we did is Mackenzie said, okay, let's dock it against the human pocket tone, if you want to call it that, and then take the top 10 for each one of them and create a heat map. And we didn't know what we're going to find. And what we found is that there's some proteins that are predicted to interact with all of these very, very tightly. And there's some that you can't see the pattern that are really differentially binding to the pHs that are really toxic, have a different population of predictors. This is one example. So we, again, these are based on human, right? Because the zebrafish pocket tone predictions, we're working on it. It's not done yet. So we're going to go from human because we're trying to use a zebrafish model for human health anyhow. So let's start with humans. And then what we do is we then take the targets identified, the human interactions, look for the orthologs in zebrafish, do the AlphaFold predictions of the zebrafish protein, see if those interactions are still favorable. And then if they are, then we're doing a species read across basically. And that's what we did here. So there's actually a crystal structure for this particular protein, a human, and we did predict it in zebrafish. And sure enough, the interactions are really favorable both for the human and the zebrafish. And in fact, the alpha naphthalene has a stronger predicted binding energy than even the substrate for the enzyme itself. So it's a good candidate. Of course, we're chasing that down. So what does it do? So this enzyme cleaves AMP to make adenosine, and this is a signaling molecule. So this is important in mitochondrial function. Mitochondria are very sensitive to pHs, so this is really an interesting target, and we have others that we're chasing down. But one thing I've learned, so I get excited by this stuff. I know it's overwhelming to my students, and they get freaked out on how do we experimentalize all of these predictions. And all I keep telling them is like, you have so many different data streams to prioritize your experiments. It's okay if you're wrong, but let's just get going. And so we can actually do these functional tests in vivo pretty rapidly now. That's what we're focusing our efforts. Last, I want to talk a little bit. I know there's a lot of PFAS activity here. The same rationale is we're trying to put in a single system that can compare the structure response to lots of different structurally diverse PFAS. So everyone knows that these are a widespread problem globally, but I still think there's a major gap of predicting which PFASs are the most problematic, if they're all problematic, and how we should prioritize our regulatory policies and our cleanup efforts. And I think it should be driven by biology, not entirely by chemistry. Even though they're related. So again, we wanted to get as many PFASs as we can get, verified standards, and then test them all across concentration like I described in this system, and then see whether or not they all behave the same way. This is a simple example a couple years ago. So at this time, everybody was binning all PFAS together, right? And it was just obvious you can't do that. And with a chain length and variation, and then the head group. So I said, let's just do a paper to demonstrate that you can't do that. So I had one of my students give me four chain PFAS. The only variability is this head group. If everybody's right, if we do our studies carefully and rigorously, we're going to get similar activities with all of these. Did them all blind, again, screen team. And we limit our concentration between 0 and 100 micromole for all of our studies. And then we did transcriptomics, and we did the internal dosimetry as well. First, right out of the gate, we ran all of these across that concentration range, and only one of them, this FBSA, produced an adverse response. These did not, a little bit of behavioral response to these three, but we didn't see anything. Certainly there's charge that could be playing a role here, but they all had 4 carbon, fully fluorinated carbons, but the response was different. So that was an interesting observation right out of the gate. So then we said for transcriptomics, we do this EC80, the concentration anchored to 80% response. So we picked that concentration, determined that concentration for FBSA, and then did transcriptomics at that same concentration with the other ones, with the assumption that dosimetry is the same. And then again, it's phenotypically anchored, same thing, exposures, RNA isolation in 48 hours. And we always have a whole back plate to make sure the animals would have had the response if we let them go, and they did. And this, we repeatedly see this scenario. So only the compound that produced an adverse effect produced a transcriptional signature at 48 hours, a massive transcriptional signature, 2,000 transcriptional changes. Very, very few. These animals were exposed, at least in the water. I'll get to that in a second. And nothing happened. Right? So maybe there was no exposure, or maybe these compounds actually behave differently.
Fire truckers.
Really?
That is fire truck.
Ice cream truck.
So when we dive into those transcription change for that particular PFAS, we see some of the actors that, fatty acid metabolism, et cetera. So clearly that has been impacted. So the easiest explanation is that, it's dosimetry. So when we actually did this, so when we measure the uptake using the exact same nominal concentration, so the FBSA and the PFBS, this one's inactive, so only no activity, only 5 DEGs, but really the same concentration of tissue concentration. May have gone to a different place in the animal. That's still a possibility. So this animal is exposed, took up the chemical, and nothing happened. Whereas FBSA, similar level exposure and disrupts development, causes mortality, et cetera. So the point of this paper, when I did it, it was like, just proceed cautiously in how you bin these things, and not just based on carbon length, which at the time was the dominant. Things are getting a little bit more sophisticated now. So kind of the things that we're focusing right now. So we have many contracts and many large libraries and drug libraries that we're doing. So we'll produce that data for lots of decision making. But really, dosimetry, I think, is the biggest thing that we need to solve. Typically, we only know the nominal water concentration, and that's not very useful when you start comparing data between systems. So we have one dosimetry model for pHs that we developed. We're working with a company right now to do a broad chemical space that we hope will be a good model that can predict tissue dose from a nominal water concentration across developmental time. So being able to predict the uptake that. So which one of these outcomes are really relevant for exposures that humans are experiencing, right? Zebrafish are metabolically competent. This is so important in terms of interpreting, even though the metabolism isn't the same exactly as humans, but showing that these molecules can be acted upon to produce an adverse effect is something that so much is lacking in these simpler systems, which is really interesting. And so as my career progressed, all the reason people would not accept in vitro data was that they'd say, oh, but you don't, metabolism. Now they just like ignore it. It's amazing. So we have to keep reminding them that metabolism is really important. Genetics, again, I think the docking that we're doing and others, I think we're going to be at a point to know when genetics matters, and more importantly, when you can predict it. So it'll be maybe in your lifetime over there in that side of the room. I'm really confident that these systems approaches, when we start having biology with the understanding like physics has for their field, we will be able to predict these outcomes in a quantitative way. We certainly aren't there yet, but I think most of us can imagine it now, right? If we know what every molecule does in a biological system, and what would happen if we modulate this by 10% and this by 15%, and I think we're getting there. We have a number of we call them secondary assays for these phenotypic screens. We're automating these right now, so hopefully we can speed up and have the cost down more. And there's been a lot of political pressure in some countries, like, so is zebrafish new approach method? Is it a non-animal model? And depending on where you are, the answer is yes or no. And some people put a window of guidance or regulation at 5 days, some put it at 3 days, some put it at 0 days. So we want to make sure that we're ready to be able to exploit what this model can bring when it's also legally allowed to do so. So we're starting to stay ahead of that. With that, if you any questions now, or you can reach out at any time, happy to answer questions.
The clinical talk is always just really next level. A little bit more in the data. I was looking at your naphthalene data, and I noticed that one hit that came across was the folate receptor. And given folate's tied to development
Yeah,
and in prevention, you know where I'm headed.
yeah, it's funny though, but if you do this, that seems to be a really sticky receptor from these predictions. So if you look at a lot of the PFAS docking data, same targets come up. And it could be real, or it could be an artifact of these docking data. So that's one we're looking at.
Given that there are studies out there demonstrating that folic acid can actually inhibit the neurotoxic effects, developmental effects. So I just found that really interesting as a underpinning of a demonstration of a mechanistic demonstration within these models that translate to mammals or others.
Exactly.
So that's really cool.
Yeah, no, exactly. And that gets to the point of trying to do the confirmatory studies, but it's actually challenging, right? So these docking, they don't predict agonism or antagonism. They're just interaction, right? So what do? Do you knock the gene out? Do you overexpress the gene? Or do you have chemical inhibitors or activators? And that's how we would go after the folate one. You try to compete with more of the agonist to see whether you can.
And given that folic acid is metabolized by 1E2.
That's right.
Yeah, that's cool.
So yeah, it was a great talk. I was wondering if, so you did RNA-seq and this is an outbred strain, so it seems like you could mine the genetic data from the RNA-seq, like look at the extremes, those that are really sensitive to a chemical and those that are not, and find
We have a
and
separate.
expression levels and so on.
The way we take advantage of those genetic background is we mined all of our concentration response data for chemicals, and we asked for chemicals that have this really nice, curve that the toxicologists say you have to have. Because again, I'm not a toxicologist, and people are teaching me what an EC50 was. That's EC50, right? You do the exposure, and at that concentration, half the animals are normal and half of them are dead. Like, how is that possible, right? And they say, oh, it's just, that's the way it is, right? Is it? So when you start controlling the biology and the exposure and the experimental conditions, and you get that response versus some that do this or some that do this, we ask the question, is there something about susceptibility in our population at that concentration? Are we detecting differential sensitivity chemical? That was the question. So we mined our data, found some good chemicals, good candidates that were relevant. And then we did those studies hundreds and hundreds of times. And then at that EC50 value, we sorted the animals, the responders and the non-responders. And we sequenced their entire genomes, like 250 of these, 250 of these, huge informatic challenge. With the hypothesis that the non-responsive ones are going to be more similar to each other and the responsive, and that's the case. And then look for like Manhattan plot type analysis. And we've identified susceptibility genes or regions in our population that explains the differential sensitivity. That's how we're attacking this model. But certainly, we do a lot of analysis of the transcriptome to try to connect it, in this case, like to the docking data. For example, is there any connection between any of these predicted interactions that might explain the transcriptional signature? That also might explain phenotypes. So that's kind of how we're trying to move through that space.
Yeah, I was thinking you could use that for your docosome experiment, where you look for SNPs in your population that affect the presumed docking site and use that as an additional screening criteria to.
The number of SNPs in this population was mind-blowing.
Yeah.
I was really surprised. I shouldn't have been, but again, they're more diverse than like the human population. The number of SNPs. So there's a lot going on.
Hi, Robyn, it was beautiful. I just wanted to ask about the behavior assays you guys have there.
Yeah.
It seems it's only few, one or two basically. So aren't you not missing a big difference?
Absolutely. Yeah, so it's a great question. So in early life stages, you can't really do complex behavior. So these motor response is what's amenable to higher throughput. There's some predation that you can do, some assays. So to get around that, I didn't talk about it, we have an adult behavioral assessment room. We built a bunch of instruments there too. So where we can look at social interactions, we can look at fear responses, learning, memory, anxiety, if you want to call it that. And so we could probe those. We published a few of those papers. And so that's a really fun part of the lab, more for these grow out studies. So we do the early embryonic insulted chemicals, and then we raise them up and then see whether we have an effect on brain development or function. But even that takes too long, right? So what we've done is we've shown that most of those types of complex behaviors, you can do them as early as 28 days. So at that point, they're pretty small. So we can put those in these, we've made these 3D printed inserts put into our zebra boxes. So we can measure those same types of behaviors as early as 28 days.
Did you try to make a prediction model of how these behaviors will change if you start from the earliest steps of behaviors, whether
Yeah.
they, that would be really cool,
It
by
would
the
be-
way,
it would be
to
good, yeah.
use this machinery.
Yeah, exactly.
So the part of the model is the outbreeding of the strains. Is the reproductive success of each part of the strain the same? Do you have to, put your thumb on the scale to keep the same level of variance in the model?
Yeah, in turn, it's a huge problem in people who run zebrafish facilities. If they aren't real careful of the way they propagate the next generation, you get genetic conversion really, really fast, just like Drosophila, same, right? So we have a spawning strategy that we do to ensure lots of contributions to the generation. And most labs don't have that discipline, and then they contact us and say, our colony has crashed, because all these recessive alleles have accumulated and the animals don't spawn anymore, like you said. And we've been able, and it's holding steady. I mean, we have a cryopreservation process in the lab too, so we're maintaining other generations just in case that happens. But no, these guys are really robust because of their genetically diverse. That's actually why we did it originally. We weren't smart thinking, we want genetic diversity. We wanted animals that would create a ton of embryos that are never going to limit our experiments. And then we thought, it also has these other attributes that are valuable.
Hey, Robyn, thanks for visiting us. Very impressive. So can you talk a little bit about your strategy for automating the transcriptomic side of things?
Yeah.
I missed that.
Yeah, so we've done that and we're doing it. We've done it one way. So we have Kingfisher system for magnetic separation. But even before that, we had to automate the way we dissociate the embryos. So we actually call it Darth Vader. So we use a bead blender because that works really well. But bead blender works better in tubes. And so we bead blend in a plate, so a deep well plate. So individual embryos, there's that long story, but we can take individual embryos, put in this deep well plate, and then we can dissociate the entire plate. And then that plate supernate and goes to the Kingfisher and isolate the RNA. We have been working with BioSpider, some of you probably know, tempo sequencing. I've been reluctant to use that platform because it's representative sequencing, if you know what that is. So basically you use 2 primers for every target. It's like a super PCR in a way. 2 primers that bind and they have to be ligated together. And then in the sample prep for Illumina sequencing, you're just sequencing just those transcripts, not all those other reads. So the cost of sample prep is really, really low. You can do multiplex the whole plate in it. But you're only going to get data on those, in the zebrafish, there's 3,600 probes. They do have a full rat and mouse and human. So it does drive the cost down. But we now know, so the reason those got selected from Broad Institute years ago said for the toxicologists, if you mined all of the transcriptomic data from the toxicology and cancer field, then you could find relationships between all the transcripts that are dysregulated. And then you could print, if you just measured a few of them, that means you've sampled everything that would have been affected if you measured everything. So it's like a leap of faith in bioinformatics. But But you can now extrapolate. So we've done this. So you can take, if you just measure, so we take a full transcriptomic data set and then measure all of them. And then get rid of everything except the ones you could have measured using this representative sequencing. And then extrapolate to see if you would have predicted using these tools, the entire transcriptome. And it's actually pretty good. So we're rethinking the way we spent all that money on it gets expensive. I mean, if there's no labor cost anymore, but there's reagent costs, that we now can dissociate directly an embryo without quantifying. That's actually the most expensive part now, is quantifying the RNA in sample prep. Just crazy. So to confidently measure low quantity, you could just take an embryo and reproducibly dissociate it. And then by volume, take that and do a sample prep, and you're good to go for sequencing. Within 6 months, I think that's where we're going to be. So what we've been doing for all of our screening, we used to do our data, come back and then grab the chemical back out of the freezer, repeat the study, do transcriptomics, and like that's kind of crazy. So now what we're doing is during the screening where we have extra embryos and we're sampling at every concentration at 48 hours and freezing it. And then we use our LIMS system to identify where in that concentration was the EC80 value. We go back to those plates and then we isolate the RNA and then we send it off for sequencing. So, yeah.
So, nice talk. In your PFAS study, did you use mixtures? And if you use, what objective you had to pick the mixtures ratios?
We have not done mixtures yet. We've done like 260 individual PFAS. So the mixtures that we would do would be the ones that have been detected, in tissues or in the environment. So what we're trying to do is try to put a number on every PFAS, a response value, kind of like a QSAR approach. And we're not the only ones. We've shown that maybe 40% of those have no biological activity, and it's not explained by dosimetry. These molecules do different things, and honestly, they will do different things in different systems. And that's something else that I don't think is appreciated in the PFAS world. You'll say, well, it affects the vaccine that in that system. That's probably true, but that same PFAS in a different context may interact. Well, we know it interacts with lots of different proteins. PPAR, lots of energy. No doubt that some of these interact with PPAR, but they don't interact with just PPAR. And not all of them interact in such a favorable way. So I think it's going to take a lot of systematic data across chemical space to start understanding the structure response relationship to those chemicals. And I think we have to keep pushing to do that. So it helps regulate, but also helps the manufacturers make better compounds.
In your susceptibility answer that you had, so how do you determine the susceptibility? Is it the dose response that you see, or is it the pharmacodynamic? Is it the kinetic or dynamic?
Right now, it's the concentration response. Okay. Yeah, but within that means like you could have different animals giving you information across that concentration response. It just tells you that there could be differential susceptibility in your population. And when it comes to do the molecular work, is actually really difficult because you can't do that work in a heterogeneous population because you can't even knock out a gene. If there's a SNP to the question, if you have a SNP at the guide site in your susceptible population, your CRISPR is going to fail. So we jump back to, we find the phenomenon and then we jump back to these more inbred lines for the mechanistic work.
May I ask a question online? Robyn, can you hear me?
Yes, I can.
Yeah, this is a great talk. I definitely learned a lot. So I'm very interested in the beginning part you mentioned about the toxicology. We've been talking about, new approach method, replacing high costly animal studies, for drug screening, for example, or toxicological assessment. So do you know any study and ever like a systematically compare, for example, how well using zebrafish for toxicological assessment for whatever medical drugs or environmental stressors?
Yeah, it's a good question. We've been doing this for years. So like the simplest one, when I started this model, is more for D&T and teratogenicity screens. And that correlation is really high. It's 80-some%. And we and many other people have shown libraries with the NTP and some companies. So pretty strong correlation. I was going to show a study. We just published this large antiviral screen that we did, and 403 antiviral compounds did it during the pandemic. And the predictivity, not only is this identifying toxic, but the predictivity of safety was like 90%. And then the predicting the failed drugs, really, really high. And the correlation with this human automated cell system was actually very high as well. So even in the drugs where you have really antivirals is a good example. We don't really have good targets in the human for the toxicity, right? Trying to hit a virus in the human. Really valuable to help sort the likely safe and the potentially toxic. There have been many. The problem is when you do these types of studies, and that's happening in all the organoid development as well, it's cherry picking, right? You'll grab 10 knowns toxic and 10 known and say, and they all seem to hit at like 80% or something. But it holds up very, 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 very well, even though it's a simpler system.
Great, thanks.
I think more work in terms of communicating that story needs to happen in this field, because the knee-jerk reaction is it's a fish. How could it possibly be useful for human? And I would say, well, it's a collection of simple systems in a plastic dish. That concerns me much, much more than the small genetic differences between human and fish versus the complexity collapse that happens when you simplify a system. Again, I think it's great for if you're trying to predict a particular effect, like cardiotox, for example. There's pretty good systems to predict cardiotox. That's not the only decision that a manufacturer has to make. Right? You also want to know that. So that may be a late decision in some preclinical testing that could, rule out a chemical. But imagine if you could do whole animal responses to your agent on day one when you have, say, 10,000 lead compounds or 1,000. And then you have another data stream that makes you think about which one you might move forward. That's the type of data that they're not ready to deal with that yet because it's so different. But I don't know why, if we drive the cost down, why wouldn't somebody just take a peek? Because you can do these studies for a 5-day full in vivo study across concentration with thousands of animals, less than $1,500, right? Crazy. Great question though.
Thanks.
So can I ask a big picture question? So you, on one of your first slides, you mentioned reproducibility.
Yep.
And so I was just thinking, how do you think about that when you've created this quite impressive behemoth testing paradigm, but in a population of animals that is unique to your group?
Yeah, well, we don't just have a wild type. We have wild type strains. We have like 70 strains of fish. So I think reproducibility has to be achieved in any system if you're getting. So we're actually, our lab is leading a systematic review right now, trying to understand how well are zebrafish researchers capturing and defining and explaining their experimental parameters in these studies, because that is where this question of reproducibility, I think, is largely coming from. It's funny, like if you were in a mammalian space, you're doing guideline studies with rodent studies, pretty prescribed exactly how you do it. And then we expect that. But in a zebrafish world, if you don't work in the field, you say, you use zebrafish, you use this chemical. So many things in between those that could impact the outcome. What was the volume? What was the container plastic? And what's the concentration when with lights on or off? And all of that are variables that will impact metabolism, uptake, and all of that is not being well captured. So the problem is that people start seeing disagreements between this phenol A in this lab versus this lab. And we've been able, a lot of what we've automated is our understanding of the importance of those parameters. That's why we locked them down. And we've done studies where we changed one of those parameters systematically and showed the impact on the concentration response. It does not change your decision on a hit, but it absolutely changes the concentration response. And that's where a lot of the uncertainty is. It's like at 1 micromolar in this lab and it's 10 in this lab or something. And we just gotta clean that up.